Hello everyone, my name is Abigahill Simon, a fourth-year food science major, psychology minor from Fort Lauderdale, Florida, currently attending the illustrious Florida A&M University. Although currently I am an undergraduate student, my long-term career goals consist of becoming a medical laboratory scientist, focusing on research in neurology and in psychopathology. For the past 2+ years, I've had the privilege of working under Dr. Violetta Silova, a renowned professor at Florida A&M University's Agriculture Department, and the lab director and chief of FAMU's Viticulture and Small Fruit Research Center. And it's due to her that I have the privilege of presenting to you a microbiomic analysis of Muscadina, rotundifolia, and vitis species using QPCR. In long-term short, we analyzed commercial grape cultivars grown in Florida, as well as in the southern eastern part of the United States. Many of you may or may not know this, but Florida ranks among the top 5 states to produce and consume grapes and grape products. These commodities consist of jams, jellies, juices, sodas, and my personal favorite, wine. Now, when we look at the population of Florida, the majority of the population consists of tourists and retired individuals. When we look at the numbers and how high they are, it makes sense why the production and the consumption of these commodities are so high. Agricultural personnel step in to analyze several grape crops from a molecular diagnostic perspective, facilitating comprehension, maintenance, and expansion of various grapevines or grape crops. The first phase of our research is RNA extraction. Researchers isolate and extract RNA from petrol tissues. Yes, it is true, several plant pathologists like to extract RNA from dormant tissues. However, within our research, we have found that an abundance of RNA is found in the petrol tissues of our grapevines. We use several protocols to analyze and characterize the several diseases that influence our plant species that are related to Muscadina, rotundifolia, and vitis vinifera. Our RNA extraction protocols consist of solution-based columns that are used to basically isolate our RNA and take it out of its cell tissues. Now, when we expose RNA in an uncomfortable environment, it's very easy for RNA to denature. So what do? Our protocols consist of several washing and centrifuging parts of our procedures, which allow RNA to be isolated and exposed outside of the cell tissues within the petrols that we've extracted. We discard several contaminants that consist of carbohydrates, lipids, starch, and several parts of cell tissues that may come in the form of a pellet within our columns, and we discard that part to make sure we keep RNA in its purified state. Once we elute the RNA, we place it in a minus 80 degrees Celsius freezer to make sure we keep our RNA in its purified state, so that although it's exposed outside of its cells, outside of multiple cells, we keep it fresh and we keep it pure to make sure it does not denature. The second phase of our research is QPCR. What is QPCR? QPCR is a molecular photocopying method popularly used in molecular biologies to detect and make several copies of specific DNA or RNA segments. We use QPCR, as I said before, to analyze and detect the several diseases that may be present within our plant species. Now, the process of QPCR contains 3 steps: denaturing, annealing, and extending. Within the first part, or the first stage of PCR, denaturing is when the temperatures within our machine that you can see here heightens itself to unzip the RNA biomolecule so that each nucleotide base is exposed in its entirety. Now, within RNA, our nucleotide bases consist of cytosine, guanine, uracil, and adenine. Now, the second stage of QPCR is annealing. Annealing is when the buffer is used to maintain pH, and this is where the temperature is cooled, so that although we have now unzipped the RNA, more specifically, each nucleotide is getting its own space so it can be seen more clearly when extending happens. Extending is the process when in which each gene sequence is copied over, a particular gene sequence is copied over and over over and over again. Now, for qPCR to occur, you need 5 imperative ingredients, which consist of the enzyme, which is more prominent in the second stage and the third stage of qPCR that keeps the process going quickly. The second most important ingredient of qPCR is the assay, or the several viruses that you see to the left of your screen. For example, grapevine lethal 1, grapevine lethal 2, GVB, GVA, and red blotch, which are the several diseases of the plant species that we're looking to detect within our sample. The third most important ingredient is the template, whether you're looking at RNA or DNA. Since RNA is the particular biomolecule that exposes a particular disease or a possible disease within our sample, we tend to look at RNA. DNA is more for gene expression, and although it can be used, our central focus is RNA. The buffer, as I said to you before, keeps the pH of our sample at a certain level. So again, although our RNA is out of its comfort zone, we keep that pH at a certain level to make sure it doesn't nurture. And one of our most important ingredients, again, is H2O. It keeps balance within our sample and also keeps RNA from denaturing. Now let's look at our results, the fun part. Now, exactly what do we get from doing qPCR? Exactly what do we yield as results from PCR? Our qPCR results is presented to us in the form of an amplification plot. Our amplification plot shows us whether there's a presence of the disease or whether there isn't a presence of the disease. If we look at our first sample in figure 8, amplification plot of sample B21212 indicates the detection of virus lethal 2. Now, what exactly are we looking at? We are looking at an amplification plot where we see 3 grand amplifications of the same sample. We copy our samples 3 times within our 96 well plate to allow consistency within each result that we get. It is very obvious, like I said before, we see 3 amplifications, which means all 3 wells within our 96 well plate showed signs of LR2 virus. Now, we are able to detect this by saying that at between the second or fourth cycle of our qPCR, we were able to detect an amplification. And this cycle went on until the 40th cycle, where we saw that although it started at the second one and ended at the 40th cycle, there was consistent amplification within our sample. Continuing on in figure 9, we are able to see an amplification plot of sample B21221, which indicates detection of, once again, LR2 virus. It's very obvious that between the 4th and 8th cycle, now these cycles, or these particular cycles at which amplification begins to show, is known to be our CT number. Now we can say our CT number is between 8 or 4, where we see consistent amplification within all 3 samples or all 3 wells of our B21221 sample. Now, a question that you may probably ask is, what if we see only 2 amplifications within a plot? What if we only see 1? Well, for example, in figure 10, amplification plot of sample 047162 indicates detection of a virus of our GVB virus. Now, as we see here, we only see 2 amplifications. They're not even huge amplifications. Our CT numbers consist to be probably between 4 and 8, or 2 to 4, where we see a grand or somewhat grand amplification in our first well, somewhat of an inconsistent amplification in our second well, and our third well is nowhere to be found. Or if we go over to figure 11, where we see amplification of a sample 047141, again detecting GVB virus, we only see 1 amplification. It's a mere amplification within our plot. Now, these are 2 great examples of the reason why, as scientists, we do the same procedure, we run the same protocol over and over again. When we see amplifications like this, it's not that we don't believe the machine, but we want to be assured and confident that there is a presence of a virus there. Although man makes machines, we also make mistakes. So redoing these protocols, redoing these procedures will possibly allow us to see amplification plots such as those that we see in figure 8 and in figure 9.
La prochaine étape in French, it means what's next. As we all know, COVID-19 shut down a majority of the work undergraduate researchers were doing, not only within our lab at the Viticulture Center, but at other campuses all over the United States. Dr. Silova took initiative and said, let's go back to the Stone Age. Let's go back to reading articles. Let's go back to reading journals since we can't meet face to face and see exactly what's going on out there. What's the different thing in terms of technology? What's hot out there? And we found that these two different articles caught our attention in regards to how we can expand our research, how we can go into the next level of technology within our own laboratory. As you can see within our first article, high throughput sequencing and the viral mix study of grapevine leaves. Within our article, we saw that researchers took 11 grapevine samples and 6 viroids, which are small pieces of RNA that contain large amounts of viruses. They're known as viroids, small bits of viruses that you can find within RNA. Our researchers took 11 grapevine samples and 6 viroids and used different types of techniques, QPCR, real-time PCR or quantitative PCR to go ahead and analyze these different viruses. Now, when they used high throughput sequencing, which is a new generation type of method to analyze certain gene sequences, they not only were able to detect the viruses that they were aiming to detect, they also found a new member of the virus, the thymomorales member of a virus that they had already were analyzing. Now, high throughput sequencing puts us at an advantage to not only look for present viruses that we think may be present within our samples, but viruses that we may not ever heard of or may not be familiar with. High throughput sequencing has not only been prominent within the States, has already been prominent internationally. As we look at our second article, high throughput sequencing applied for the identification of viruses infecting grapevines in Brazil. These researchers in Brazil took 17 grapevines and used high throughput sequencing methods to go ahead and analyze their different viruses. And once again, not only were they able to detect the viruses that were present within their samples, they found new members, new types of viruses that may be detrimental to the growth of grape crops and grapevines. Now, what does this say for us? This puts us, as I said before, at an advantage to look at the several different types of viruses or diseases that may be allowing or detrimenting the several grapevines that we have at our research center, or the several grapevines that other grape growers and plant breeders may be experiencing. Now, in relations to COVID-19, which is a disease that has taken the world by storm, they are also using these several methods that we see here, such as QPCR, to analyze and detect whether COVID-19 is there. They're not just taking cotton swabs up our nose for no reason. They're not just taking cheek swabs out of our mouth for no reason. They are extracting the RNA biomolecule using QPCR to detect the several gene sequences that may be present within RNA. As I said before, RNA is the molecule that exposes several viruses, possible viruses that may be present within samples, human samples, or plant samples. And we thought that it was very, very interesting how we researched that different health personnel were able to use qPCR to detect these viruses. I want to thank, in conclusion, I do want to thank Dr. Violeta Silova, who I've had the privilege of working under for the past 2+ years